Welcome back, everybody. In our last talk, we learned about the endomembrane system. And by doing so, you learned about more than 50% of the major organelles inside the eukaryotic cell. But now I want to finish up our picture of the anatomy of the eukaryotic cell. So let's get started here. The first two organelles we're going to look at are involved with the conversion of energy from one form to another, and they are mitochondria and chloroplasts. Mitochondria, many of you know as the power plants of the cell, but what does that really mean? Well, what it means is that they produce ATP, and ATP is the energy currency of the cell. In other words, it carries a small amount of energy that is useful for causing the chemical transitions that occur within a cell to occur. And mitochondria create ATP by breaking down the energy-rich molecules in the cell, such as glucose or other carbon-based molecules, through the process that you already know called cellular respiration. Chloroplasts, which are found only in plants and algae, are the sites of photosynthesis. And in photosynthesis, you will remember that light energy is captured and stored as glucose and other carbon-based molecules. Now, mitochondria and chloroplasts are not part of the endomembrane system. And what's most interesting about them is they reproduce themselves and they have their own genome. And what's even more fascinating about that is that the genetic material in them is not eukaryotic DNA, but actually looks like prokaryotic DNA. They actually transcribe and translate that DNA and make many of their own proteins that they need to reproduce themselves. This has caused people to hypothesize that both the chloroplast and the mitochondrion were once free-living bacteria. And through mutualistic symbiosis, these once free-living bacteria were taken into a cell, and that is a major part of the story of the evolution of eukaryotic cells from prokaryotic cells. Let's now look at mitochondria. Mitochondria are found in almost all eukaryotic cells. They have two membranes, an outer smooth membrane, and then an inner membrane that contains many folds, and the folds are known as cristae. And what I'm going to do here is first describe it to you, and then I'm going to show you a picture. So we'll go over all this twice. The inner membrane creates two compartments inside the mitochondria. The first is the intermembrane space. It means that's the space between the two membranes. And the second is the space inside the inner membrane, which is known as the mitochondrial matrix. The cristae, these folds in the inner membrane, create a huge amount of surface area on the inside of the mitochondria, and this is the site of many of the enzymes that are necessary in the synthesis of ATP. And we'll learn about that in great detail sometime in the spring when we get to study cellular respiration. Here we have the outer membrane and the inner membrane. So between them is the intermembrane space. And you will notice that the inner membrane is folded inwards. This is what we mean. These are called the cristae. And then inside the mitochondrial matrix, which is this liquid substance on the inside of the mitochondrion, are ribosomes that are floating around, and as well as circular mitochondrial DNA. This is what a mitochondrion looks like when you see it in an electron micrograph. And you can see the cristae poking in, and you can see the ribosomes. You cannot see the DNA in this micrograph. So that is the anatomy of a mitochondrion. I highly recommend that you pause this video and draw and label a diagram of the mitochondrion for yourself. So now let's move on to chloroplasts. Chloroplasts capture light energy and take that energy and put it inside the chemical bonds of organic molecules such as glucose. Like the mitochondrion, chloroplasts have two membranes. But within the chloroplasts are disc-like structures called thylakoids, and they are membranous and they are filled with a fluid inside, and they're stacked on top of each other. I'll show you a picture in a second. The thylakoid wall contains the green pigment chlorophyll. Remember, chlorophyll is the pigment, the molecule that is able to capture the energy in sunlight. That energy can then be used to synthesize glucose from carbon dioxide. Anyway, the thylakoid wall contains this pigment as well as other enzymes and other molecules that function in the process of photosynthesis. So much of photosynthesis takes place inside the thylakoid, and we will study this in great detail later in the spring when we study cellular respiration. The fluid inside the chloroplast but outside the thylakoids is called the stroma, and chloroplasts are found anywhere that there's a green part of a plant. So here's a picture of the chloroplast. Notice two membrane layers, and then inside these green disc-like thylakoids that are stacked on top of each other and are connected through little passageways. Each stack is called a granum, and as I said earlier, in the walls of this membrane are many important proteins for the process of photosynthesis. Also inside the stroma, which is the fluid that surrounds the granum, are free-floating ribosomes and then chloroplast DNA, which is circular and prokaryotic. Here's what we can see in a micrograph. Here are the stacks of thylakoids, the so-called grana, and there are the passageways connecting them. And you can see a few ribosomes in here as well. So those are the two organelles that have to deal with energy transfer, chloroplasts and mitochondria. Let's move on and look at some other structures inside the cell. Cells are not just a membranous sac of a cytoplasmic solution. They also have an internal skeletal network, but it's not a rigid skeleton such as you and I have, but it is a skeleton of flexible proteinaceous fibers. And it is a dynamic network of fibers that extend throughout the cell. And the cytoskeletal elements are classified in different ways. And we're not going to really talk much about them, although we'll talk about microtubules. But you're not going to have to know the criteria for each of the different types of cytoskeleton. And the cytoskeleton basically organizes the cell's structures and activities and anchors many of the organelles in place. It helps support the cell and maintain its shape, and it also provides tracks for motor proteins to produce movement inside of cells. So, for example, when we see vesicles moving through a cell, they're actually traveling on the back of a motor protein, which is attached to the monorail of a piece of cytoskeleton. And I'll show you an illustration of this in a second. So here is what the cytoskeleton looks like when we see it in a micrograph. And here you can see a microtubule has been pointed out, and another element called a microfilament has been pointed out. And here it is in the artist's representation. And notice that it is associated with the centrosome, which contains these two barrel-like objects called the centrioles. And we'll talk more about that in a second. And then here is a microtubule to which a motor protein has been attached. And this motor protein is attached to a vesicle. And basically what happens is the motor protein has one shape, and then if it uses some ATP, this leg will move forward and attach to the microtubule up here, and then it will relax and this other leg will move forward, and it basically walks itself and the vesicle it's carrying along a microtubule as if it's on a monorail. Here's a micrograph of a microtubule as well as a vesicle, and under here somewhere is this motor protein carrying the vesicle along the microtubule. Next, I want to talk about centrosomes. They're found only in animal cells. They're associated with cell division, and they are also the microtubule organizing centers, microtubules being one of the types of cytoskeleton. And I'm not making this up. The microtubule organizing centers are abbreviated as MTOCs, MTOCs, and these are important during mitosis, and mitosis is the process by which replicated chromosomes are divided during cell division. The centrosomes contain the centrioles, and I will show you a picture of the centrosome in a second. They duplicate and migrate to the opposite poles of the cell before cell division, and then they're involved with the separation of the genetic material to each of the new daughter cells. Here's a picture of the centrosome, and you can see here's where it's located in the cell. Here are the two centrioles, and the centrioles themselves are composed of a cytoskeleton element called a microtubule. that occur in triplets, and then they're connected by other fibers. Next, I want to talk about the cell wall. This is found only in plants, fungi, and in some prokaryotes. The cell wall is extracellular, meaning it occurs outside of the cell membrane, and it's what distinguishes plant cells from animal cells and fungal cells from animal cells. The cell wall protects the plant cell, it helps it maintain its shape, and it prevents excessive uptake of water. And so you want to think about that. How does the cell wall prevent excessive uptake of water? If water is flowing into a cell, the pressure inside that cell is going to increase. The cell walls mean that the pressure will increase even more. That's because the membrane can't expand infinitely. So the cell walls increase the pressure. At some point, the rate of outflow due to the pressure and the rate of inflow due to a process called osmosis that we'll talk about in an upcoming video, those two rates are equalized and the cell has no more net gain of water. Plant cell walls are made of a sugar compound called cellulose, and the cell wall is more like the Eiffel Tower. I don't know if you've ever seen it, but the steel is not solid. There are holes in it. It is strong, but it is perforated, is the idea of the cell wall. Plant cell walls also have multiple layers, and if you ever take a botany course or an advanced biology course, you might learn about them in more detail. Let me show you a micrograph. Here it is, and here you can see some of the different layers in here. And then look at this. Here's this whole cell wall, and then this little dark line on each side here, and here is the plasma membrane or the cell membrane. And here's our artist's rendition of it. We have the plasma membrane on the inside, and then the plant cell walls, and then the cytoplasmic solution inside. Finally, we want to talk about a weird adaptation of plants called Plasmodesmata, which are basically intercellular passageways that occur only in plants. They are channels that perforate the cell walls and connect adjacent cells to each other. And through these Plasmodesmata, water and small solutes pass from cell to cell. And here's a picture of the Plasmodesmata. So what you're seeing here is one cell and another cell, and then these connections, these passageways, so that the cytoplasm can move from one to the next. And I'll leave you to think about why it might be beneficial for plants to have Plasmodesmata. That's it for today. Today what we've learned about is the two organelles that are key to energy transformations in the cell, the mitochondria and the chloroplasts. We've also learned about the cytoskeleton and that it is proteinaceous and that it organizes and gives support and structure to the cell. We've learned about the centrosome, which organizes a lot of the cytoskeleton and is important in the separation of the genetic material during cell division or mitosis, which we'll learn about in a few weeks. And then finally, we've learned about the cell wall and the Plasmodesmata in plants. So, after you've viewed this video, you should go outside, look at nature, and ask biological questions. And I'll see you guys soon.